we started to see, okay, we have a funky pattern, isolation by distance, and it's different. There seems to be something going on with the dispersal of these guys. And so I try to do the next step. Okay, we have kind of a suspicion. How can we develop a hypothesis, and how can we test that then? Okay, so the first thing is simply what it says here, visualizing. And I follow the great example of Robin, not try to run the code. Yeah, you can run the code, and hopefully it works. We tested it 17 times, and it breaks my heart that we don't run it. But actually, it's not really necessary. The only thing I might change is make it bigger. Is that even better? Cool. Okay. This is a method which is called EEMS, and I would like to explain first what it does, and then come back to the example. Yeah, so just keep in mind what you have learned. Not sure what you have. Okay. Now this is this beautiful method. It's called estimated effective migration surfaces, and Robin kind of used it in her thesis as well. No? Okay, later. And it is actually a fairly old method, but it's actually not used very much. And I think one reason where Dart shines, it makes it easier now to use it. Yeah. And I actually almost fall in love with it because I think it's cool. And what does it do? You have your sample, yeah? And the normal approach would have been, oh, I create thousands of different landscape hypotheses, resistance layers, and I run them through, and I dredge the data, and I find some relationship, and I say, hooray. But it's probably not the right way, because normally we should do the following. We should think about hard what could be important, and then we could maybe develop the hypotheses. Yeah? And this one is helping us to develop the hypotheses. It's not the testing. So it's the first step. And they developed that. And what did they do? This is elephants in Africa. And this method is doing the following. You have your samples. And actually, they have 2 species of elephant just to make it more complicated. Doesn't matter at the beginning. Then they put a grid, a triangular grid. No reason why. Just they're mathematics, and this has a cool property I don't understand. So they put that on top, and then they look at the samples and kind of put every dot which is closest to a center here to which something they call deem. Yeah, so they summarize a little bit your samples on the grid. And then they run something which is, that's why the method is called EEMS, estimated effective migration surfaces. They say, okay, things which are on there, they should be similar, yeah? And things which are connected by some distance should be dissimilar. And that the clever thing is the dissimilarity is linked to a migration. It's called effective migration. So it's not only going from there to there, it takes also, because the genetic data drift and also other structure thing in mount. So that's why it's called effective migration. It has all these things what could make you different, not only distance. So in the end, it does nothing else than try to fit this data, but it uses a plausible model in the background. A real genetic model is based also on coalescent, yoo-hoo, again. So and I like that. It's not like, there's so many interpolation surfaces, things you could run on this data, and they're all not meaningful in terms of genetics and how the system works. Yeah? And once you do that, you get this to do a little bit of interpolation to make nicer maps. Yeah? But basically, this tells me this is a log m, it's a funny system, it's a log 10 actually. Means if I have a 1, my migration rate, if you want to, is effective migration is 10 times higher than the average migration rate. So everything that's connected by blue areas is highly connecting habitat. Everything that's brownish is, barrier, yeah? So it basically tries it, and you can see, okay, this looks nice. So we can, and that's a good thing, kind of create our hypotheses, We first now can visualize what is the difference between these samples we have using this approach. I went through that. Not sure if that's too interesting. I won't stay too long. But basically, look only at the first here. That's a PCA of some data. So they simulated a barrier and no barrier. Yeah, two versions here. And basically, they wanted to show if this can detect that in a PCA, if you have a uniform, it doesn't come up nicely. This is a uniform example. Everything is the same. This is a PCA for it. Yeah. So if I have a uniform example, the PCA looks like this, because you sample left and right, and you know you still have a differentiation because they're different. And if you have a barrier, yeah, then it really nicely detects the barrier, the method. It finds it, yeah? And the PCA looks like this. And this guy says, this doesn't look too different in terms of a PCA, yeah? But this obviously, uniform versus barrier, looks very different. So it has an additional effect to understand your data when you compare to PCA. They don't say don't do it, it's just, you can see it adds the spatial interpretation to it. Yeah? Make sense? Good. Because it wasn't explained well. Okay. No, don't go there. They really nicely explain in the paper, have a read about it. There are different elephants. One is the savanna, and the other one is more to forested habitat, and they can nicely explain what's happening here. And because it's different species, they also can explain this. So they can really nicely explain the pattern, and it makes completely sense for elephants why they get this kind of surface. Okay, shall we go back to this one? Yeah. Good news is it is actually quite straightforward to run it. Maybe very quickly. This is how you run the EMS. You give it your GenLight object or Dart R object. Buffer, you know, you have to send basically which array I'm looking at. And you can give it, it's a bit of a, it's not really nicely implemented yet. You can give it a surface, say the surface of Australia. If you look at foxes, I'll show you later an example. Or if you give it a buffer, it just takes a certain distance from the, it creates basically a hull of your sample and goes out a certain distance. And that's the area it uses for estimate the migration surfaces. Yeah, so you have to give it the area, and the path is basically where your program is running. The numbers of teams, which should go further there, funny. That's an important one. It's basically how your grid, how detailed it is. Yeah, if you set it to 10,000, it's very detailed, but obviously then you see there's not many data points you get, because you know there's only single individuals at one of the teams. And understand you need both. You need many individuals at one team, but also many teams. Why? Because you want to see these ones are similar here, all of them, because they're next to each other, and the other things are further apart. So it is a bit of a, this was actually one of the exercises you can do, play with the numbers of teams and how that affects the grid. Unfortunately, this method is semi-nicely implemented. You have to run the whole thing to see how the teams are done, how the grid is set up. And so the trick is run it with very low numbers of iterations. It's a Bayesian method, I didn't mention that. So run it with very low and just look at the teams and see, oh, that looks good, and then do the full run. And that's, when I said full, they recommend something like 8 million iterations, 2 million burn-in or something like that. Look at the thing. So it runs, honestly, I had foxes, 3,000, it runs 5 days if you're unlucky. Yeah? It can. But if it's just a small example, it can be much quicker. Okay. This is not important. This is just unfortunate output. This is basically a visualization of the area it takes. And then you get 8 plots out of it. Yeah. Let's do it here. I have the plots here. It's easier. Okay. 8 plots. And the first one are the migration surfaces. Yeah, and then you get the similarity, dissimilarity surfaces, how similar are the genes, and then you get some diagnostic plots. Okay, this is the one that everyone shows in their paper, and this is the same thing. This is on a logarithmic scale, and then pronounces a little bit more, but you can see it's actually the same. It's just playing with the colors. Yeah, what does it tell us? These guys are linked very nicely because it's bluish. These guys are linked, yeah? And in between there seems to be a barrier, okay? So we get a first idea, and then you can think about, obviously look here, these are I plotted also our samples, and you can see if you have lots of samples, you have a bigger circle, means there are lots of individuals in that DEAM, and if there are less, okay? So, and if you play with the setting of your grid, you see how well this is distributed in terms of numbers, okay? Good, then let's go to the next one. This is just playing a little bit. We basically put the surface on top of our map, and you can start to see maybe rivers could be important, yeah, or something else. We are working with soil, and we will see there is actually one landscape type, namely clay, which is in the middle between those things, and that might be a barrier. Okay, that was migration rate. The other one is the Q rates. That's kind of the diversity surface. And that means what is the similarity within the DEAMs we get here, yeah, and between the DEAMs. And basically that tells me that these are similar, but those guys are more variable on the other side, yeah, in terms of their population structure. And if you remember, does it make sense? I wonder, there was male, female, so forget it. Yeah, okay. Yeah, it's funny, most people don't look at that, and I'm also not 100% sure how much information you get out of that, yeah? But you can do that. The diagnostic plots are the more important one, I think. This is the fitted dissimilarity from the model, yeah? And this is the observed dissimilarity. And obviously they should be nicely on a line, yeah? Then basically your model, which predicts certain dissimilarities, is really in line with the observed one. And you can see this is pretty good. So you can start to trust it. And then it looks also at the dissimilarity within, the fitted dissimilarity within DEAMs and dissimilarity, observed dissimilarity within DEAMs. So basically the Q plots, how well do they fit the data? And again, they should be also following a line. Yeah? And yep, looks good. Could go on. This is a simple isolation by distance plot with a bit more fancy naming. So we have seen that before, and you can see a little bit, taking obviously the DEAMs, not the individuals like we have seen before. This is based on the DEAMs, and you can see there seems to be maybe two groups or not much of a pattern. And that's okay, that's just how the data is, you know, okay? And this is just Bayesian stuff. In the end, when you run your, what's the word, iterations, your samples, you want to have a mixing of the, if you run it 3 times, what's the recommendation is, they should mix and be at the same level. In this case, they did. Make sense? Yep.
Do you have the effective sample sizes in the parameters?
The microphone. Yeah. It's good, it takes longer because I have to think about that.
There's a package will do it, so you can get it straight from that plot.
Say again?
So the effective sample sizes of the parameters in the MCMC, because that's how you know if you've run your MCMC long enough to believe that it's close enough to infinite.
Good question. You get a lot of output, but I haven't looked at that. Yeah, thanks. It makes sense. Yeah, obviously you want to, it's kind of the, what is it, the R0 value or whatever you want to have.
I'm enjoying very much both presentations about landscape genetics. No, there's no but. It's just a comment. I think that it might not be clear that for folks that, you know, a Mantel test has many issues in depicting IBD because of population structure, right? So like a partial Mantel test then can add a matrix of geographic structure that could be different than spatial distance per se, right? And fix this problem. But this seems to be more powerful, right? Or is that your view on that? Like we've played a lot with these, and you can see that you get a decay of IBD in areas of that could be migration corridors, or you have, based on the average of the migration surfaces, right? So you can identify the barriers, you can identify the corridors, but are you guys trying to come up with a solution to the issue of isolation by distance from other effects, like geographic clustering of populations, which Robin showed very clearly in that soil type barrier? Is that what it is? Is that where this is going? I was just thinking about what you are telling us about migration surfaces can help fixing the issues of IBD patterns that might be not real, that might be, impacted by structure. That's Patrick Meerman's classic criticism about using Mantel test to IBD. I don't know if you...
Yeah, so I guess we wanted to show from the start, you can run IBD, and in perfect situations you would expect that linear relationship, and then when you start to see other things influencing it, you can have a look at where those patterns of IBD change across the landscape. So that's what we're seeing here. There's, potentially a higher migration on either side. IBD is kind of acting in a consistent way on either side, and then there's this barrier in the middle where migration rates are reduced. And then the next step will be to actually directly test that, and you can include IBD as one of your variables, I guess, and kind of isolate that out. Yeah.
So, in short, I will show now basically the partial Mantel test and why we do that in some sense, and also we use the MLPE method to show, to test the things. But you're right, there could be any other ways than just geography who is affecting the population structure. And you... Pardon?
I guess my main comment is that we have to distinguish geography from spatial distance. These are different things most of the time. And that was very clear, I think, in my view, in the example that Robin gave. Geography and spatial distance are not the same thing, right?
Yeah.
Because of barriers on the landscape.
Okay, I'm totally with you, and it's actually, I think that's what I'm gonna say, but you tell me if it's not. Okay. Okay, so that was, as I said, kind of a way to visualize it. There's no test involved. It'll just say, there's some diagnostics plot, and it fits the data. So it's nice, but it's not what you actually want. Yeah? So how can we do that? And one approach, and just to let you know, there are actually quite a few more. Some of them are more esoteric than other ones. This is one way how you can test different landscapes and do they have an effect of your observed pattern of genetic distances or similarities, however you call that. Yeah? Okay. I think it's time for some cool animation. So it's called isolation by resistance. Yeah? And how is that? What's the idea about that? The idea is the following. Think about, you have four, wow, why is it 1, 2? Interesting. I thought it stops at some time. Okay, think about, we have a landscape and populations. Okay. And if the landscape doesn't have effect, they kind of migrate, and the only thing we have here is kind of this isolation by distance effect. Yeah. So they kind of become a little bit dissimilar over time because, those two are a little bit closer to each other, though on average there will be more exchange here than between this and that. Yeah? Okay, what if the landscape looks... I don't like that. Okay, it should stop, but okay, it doesn't matter. Think about the landscape is kind of a bit as if it's a barrier. Could be a different landscape type. And I know the choice of green is the worst ever possible. It should be more red or purple or some other thing. Things where the individuals from the species don't like to walk over. Yeah? and to migrate through. And then they obviously go around it, and suddenly something that looks close to each other is actually further away because you have to go around it. So that doesn't mean actually you think about individuals go really around, they're just less effective in that direction, yeah? But you can depict it like that, and if that's the case, then the pattern how dissimilar pairs of these things are kind of influenced by the landscape, yeah? Make sense? And just to explain how this is done, it's basically very similar to Robin said. You have, first of all, a pattern which, a matrix we call the G matrix, which is simply the distance, and you can use any genetic distance you're a fan of, Euclidean distance, proportion of shared alleles, or any other measure you think is a good one, to measure distances between these population or individuals. Yeah, it goes in both ways. And then we have something which is simply the Euclidean distance. It's kind of our null model, because we know things which are further apart, it's harder to get there. We calculate the distance between all the pairs, and then we find out, yeah, it follows this pattern. And if that's the case, then we have the plots Robin has shown us, and yeah, okay, we found something. But the landscape doesn't have an effect in that case. Or we calculate something which we call least cost path distances, or circuitscape, or I forgot the third one, there's some others. And the idea is, I show you only one version, that's the least cost path in the analysis, but the Dart R uses also circuitscape, and if it's too slow, you have to run actually circuitscape. The difference is, least cost path is actually what you think it is. This guy goes from here to there, knows everything about the landscape, and finds the optimal path. Yeah? It's kind of a strong assumption, but that's how it is, and it's nicely visualized. That's why I show that. Circuitscape is a little bit different. Is the green area actually a really, really deadly thing? No, you will never touch it, or is the green area just a little bit of a barrier? And so if you think about least cost paths, if this is just a little bit of a barrier, it might be cheaper even you go over there instead of going around. So it depends, these values depend really how you code your landscape. Yeah? Rob?
Sorry, maybe I'm just being really ignorant. How do I calculate the values of the final matrix?
This one?
Yeah.
You use a function.
Based on what?
Based on what you give it, basically a landscape, a raster. Do that in a sec. So give it the raster. You need that? Sorry, should have said that. Yeah. And then here, this is just coordinates, and this is coordinates. But here you have to get the landscape, and you do some GIS thingy things. Yeah. Or use Dart R. Okay. So we have three matrices, and the question is how we get them together. And if you just do G versus your cost distance, yeah, you could try to do that, but why is it not a good idea? You want to have a correlation between your cost distance and your genetic distance, yeah? But not between your genetic distance and your Euclidean distance, because that's a null model, yeah? But there's a catch. What is the catch? You could do a linear regression and done. The catch is actually, these could be highly correlated, and unfortunately quite often they are. Yeah? They're not independent from each other. And that's why you do quite often something, in one case, a part first for your Euclidean distance, because that's your null model, yeah? And if there's still a pattern, then this is actually the landscape, yeah? And the different ways to do that, and one is this partial Mantel test, which is actually fairly simple. You do a regression, G versus E and C versus E, and then regress the residuals of those two things against each other. I'm not sure if you want to explain that more. I can't actually explain it more. That's how it is. Yeah? And there is some variants of that. You do, this test of Wassermann. I explain in a second. MMR. And there is the one I didn't put it there. MLPE is the one which Robin will tell us about, how you correct for this non-independence. And the idea is, yeah, mainly there's a relationship between Euclidean and least cost distance. And we can discuss that a lot. You want to make it as independent as possible from your landscape to find a good effect. Yep. Thanks, Rob is nodding, that's good.
Only a clarification. So you said you have 3 pairwise distance matrices. Do you use all of them at the same time, or you choose one? Oh no, you use all.
Yeah, good question again. I use all of them, 3 in 1. Yep. But you can even go further, and that was the MLPE. You know, you can have only one cost distance. You can have several hypotheses. Say sand is a barrier, or water is a barrier, or something. And some people throw them all into one thing, and then they try to find out which fits best.
So basically those are your environment, landscape variables?
Yes, basically the landscape is coded in, hypotheses on the effect of the landscape is coding in this cost distances. Okay. And you can create as many as you like. I don't like that very much. I like the approach I try to set out. You plot your data, use maybe EMS to get your hypotheses, and you run maybe 2 or 3 very, very distinct hypotheses. Not what I see sometimes, which I would call data dredging. People try 17 different landscape and one fits, and that's the one. But we all know repeated tests is on the same data is dubious. Yeah.
And then the other thing, just the slide before, you said you test for the relationship between, the correlation, I guess, between your genetic distance and your cost, but not between your, what was it maybe before?
So actually what you do in a partial mental test, you do a regression, your cost distance versus Euclidean distance, yeah? You do a regression, your genetic distance against the Euclidean distance, yeah? Mm-hmm. And then you do a regression on the residuals of both of those things.
Okay, but because before I think you said you don't do, I don't know if I noted it, genetic versus Euclidean distance. Didn't you have like a cross on that? Yeah, but not G, the correlation between G and E.
Yeah, sorry.
Sorry, yeah.
Some could say we just do that, yeah. But it would ignore this bit. This is basically a raster, now a vector format from Robin. She gave that to me. These are the landscape types, and there's a bit of a GIS voodoo, and I don't go through that. What we have to do in the end, we create this kind of things, which is every landscape type. This is now a raster. If you're a GIS person, you know what the difference is. If not, it doesn't matter much. And each value, so 0 codes for the clay, whatever, 4 is tidal, and so on. So we have it kind of a raster, and it's numerical, yeah? And what we do next is, we have to recode that a little bit to make sure we have only one thing, or that's my hypothesis. I have, if you looked at it, clay could be a barrier, is my hypothesis, yeah? Because that's the big thing in the middle. Here, the white, hang on, make it, this one, yeah? That's clay, and we have seen that there may be an effect, you know, from the EMS. We thought that could be an effect. So we want to test this hypothesis. Clay has an effect on the structure, yeah? And we do that by just separating the whole thing, and then we have basically only a landscape like this. This is now coded with a resistance value as a bit of GIS voodoo, and this, I think I chose a value of 20. And you can discuss it forever, what value is a good one, yeah? It's just higher than 1. 1 is basically no resistance. 20, it's pretty costly to go through here. When here from there, it's costly because every step costs me more. So it's better to go around at some stage. Yeah? And this is water. Water is really costly. You don't go in there. Yeah? I should have made it even higher. I should have called it 500. Or missing data, if you don't have a population in there, then you're in trouble. Yeah? And we come to that when we do that. So basically, this is my hypothesis now. Yeah? How do I test that? This actually should be closed here, and you could play with it. So that's why I like least cost, and the same is here. You can see it's cheaper to cut through there. Yeah? But that's kind of the hypothesis. It's enough to avoid basically everything of the clay most of the time. Yeah? And I say, this is just an image. It's not means they're not running over the clay. Yeah? It is basically just harder and less individuals go over there. Yeah? In the end, the exchange is lower. Yeah? And then you put all the three matrices in this Wassermann if you want it. That's one of the tests people like to do. And this is a partial Mantel test. It's two. You check, is the effect of clay, which is my resistance layer, on the genetic distance, corrected for Euclidean distance. Yeah? And this is meant to be significant, hooray. Yeah? And then you have another one. You test genetic distance against Euclidean distance corrected for clay. Yeah? So the opposite. And this should not be significant. There's some people think this is not good and it's not independent and blah, blah, blah. So there's some debate about it, but that's one way you can test the effectiveness of landscapes. And because it's easy to do, I implemented it, but it's not maybe the best thing to do. Does it make sense? What I always would like to recommend, one more thing before I finish and pass on to Robin, is you plot your data, yeah? And these are your three matrices, yeah? And help me to get that correct. Which one is which? This is Euclidean distance. Hang on, this is not good, is it? Ah, no, that's genetic distance against clay, yeah? And this is genetic distance against Euclidean distance, yeah? And in this example, you can see actually, there's Euclidean distance and the cost distance are highly correlated, yeah? There's not much difference between them. That's not good. So I would not trust my result a lot, because if I have a really high correlation between clay and my Euclidean distance, it's hard to separate the effects between them. It could be either or, yeah? Some people would say, no, it's below 0.7, I can do my Mantel test, I trust it. That's kind of an often used threshold. But it's a bit like how much you believe. Yeah, but what maybe we can nicely see is this pattern here. These are the guys which have to go not around. Yeah, these are fairly far, but they're all in non-clay habitat. So they are similar than they should be, and the other one follow this one. Yeah, so you can try to find some pattern here. Make sense? Yeah.
Sorry, I was like just had it in my lap on. I was just wondering, in terms of, I guess, pure hypothesis testing, what is the justification of creating a resistance layer versus doing something like generalized dissimilarity modeling to see how different environmental factors, whether they contribute? So you can put distance and all of these other things in, all your environmental layers in at once, and then have it actually look at whether there's a relationship and it allows like a nonlinear relationship between distance and an environmental variable.
Yeah.
So I guess that's a really nice way of looking at barriers and things where you expect like a complete turnover, but it doesn't take into account the kind of distance needed to be travelled. So I think I would use that if I expected there to be like a really sharp environmental gradient and a complete turnover in genetic diversity. maybe more of a kind of isolation by environment pattern, whereas isolation by resistance is still thinking about kind of gene flow across the landscape and where it's restricted and where it's facilitated. Does that make sense, do you think? Maybe.
Okay, just to finish a little bit, obviously we could test now another hypothesis. The sand is actually good or bad or whatever you have, and you have to recode your matrices, your raster and that, and then you can calculate that the same for sand and see what kind of effect that has, yeah? If it has resistance, and then you could do a single test, or you use something which is called MLPE, which I don't talk about it, do I? There's one more thing which I like to, there's an example here, and it's actually cool because it's antikinase and Kailua, so I made that from here. So that's Kailua, and just to give you an idea, food for thought, and I have 3 things, and you have to find out, it's a simulated data set, but you have to find out which of the raster landscape is the best one. That's your task, yeah? And just to give you one more thing, this is elevation, yeah? Close to the beach, it's low. But what is more important, quite often we put in something like roads, yeah? And people don't do that right, I think, very often. If this is your road layer, yeah? Basically, this is a barrier, and everything else is not. To find an effect of a road, you have to code the resistance of the road really high, yeah? Because there are not many pixels of that color. If you model this with a 10, the animals laugh about that because it's a thin area and they just go through and it doesn't have any effect, yeah? So how you code your landscape, that's a lesson to be learned, is really very much dependent on the layer you have, yeah? If it's a linear thing like this, and it's just small, probably has to see an effect has to be really high in terms of resistance, yeah?